Okay. I think I'm frozen though a little bit. Is the quality good by you guys?
If you're looking at YouTube, it's actually 20 seconds behind what's happening on Discord. So yeah.
Okay,
but
great. Then I will go ahead and get started. Thank you very much for the kind introduction. And yeah, welcome to the seminar on Snakemake, which is my favorite piece of reproducible research software. So before we get started, I'd just like to I would just like to ask that you, if you are connected to a Linux machine or perhaps a Mac OS X machine, that you can clone this repository. So this repository contains this presentation as well as a set of example workflows, just very simple examples that I'll be working through with you today as I go through this presentation. So if you'd like to follow along, you can also then install Miniconda if you haven't done so already, using these commands, and you can then follow along with the code. So while I'll leave this open for a little bit, so you can have a chance to set this up if you haven't done so already. And I'll just tell you a quick story about how I got involved in Snakemake. So actually, after I graduated from MIT, I started working as a bioinformatician in a small biotech company in Boston called Vitro. And one of the tasks that I was assigned was to create an analysis pipeline for the set of immunosequencing data that we were generating. And I had actually used Make, which is another older pipeline software, during my grad school. But I wanted to try something a bit more user-friendly for this new job, and so I tried Snakemake. And this was actually in the early days before it became really the popular software that it is now. And so so there were still some bugs, and I was often chatting with Johannes, and he was very quickly submitting bug fixes, and I was very impressed with how hardworking he was. And eventually, it turned out that he was also in Boston at the time, and we ended up meeting up and working together a little bit. So we actually hired him as a consultant in Vitro. And then later on, when I decided to return to academia, I was looking for postdoc labs. And by this time, he had become a professor at the University of Duisburg-Essen in Germany. And he actually offered me a position. So we hired him, and then he offered a job to me. So the irony is definitely there. But yeah, we've had a good relationship, and I love the software that he's created. So now I hope you've had a bit of time to set this up if you haven't done so, and we'll get started. So, what is Snakemake? This, as I mentioned, is a software to create reproducible and also scalable workflows, so you can easily parallelize many jobs over many cores, over clusters, or over the cloud. It has a large and growing bioinformatics community, on average 3 citations per day as of last year, due to its clean syntax, so it's easy to use, and its interoperability, so you can use computing environments that make it more reproducible. It's also, in contrast to older pipelines like Make, it is very easy to quickly prototype workflows due to the fact that you can always jump into Python whenever you need to, and Python is one of the staple languages for bioinformatics, so everyone knows Python. And finally, it's now been battle tested over the years, so it's very feature rich. It has many different options to configure how you want to execute it on a cluster or on your computer, and it's very configurable. And again, It's designed by this man, who's Dr. Johannes Koester, who's now a professor. And Johannes Koester is also the lead developer of Bioconda. And so Bioconda is similarly a very easy to use package manager for a lot of bioinformatics software, and it's been embraced by the bioinformatics community, partly because it's easy to use, and partly because Johannes is also just a very welcoming person. So he is very involved in the community, and he invites people to contribute. So it's really a nice bioinformatics community that I encourage all of you to be involved with. So as I mentioned, Snakemake was originally inspired by GNU Make, which is sort of the de facto standard for compiling software. So this is historically for things like compiling using the GCC, for example, C files to create executable files. But the general idea is that you have a set of dependencies, these input files, and you want to create some output file, in particular now this myapp.elf, and this app depends on a set of executable files, which depends on some intermediate files, which then depend on the sources. So GNU Make is, in a way, very similar to writing a pipeline, because it specifies these dependencies, and it figures them out for you automatically. So so here is a very simple example of using GNU Make. So here we're saying we want to create some output file, which is hello make, from some input files, which in this case would be these two C language files. And we're going to use some command using the GCC, the C compiler. And then there are some special funny symbols that say take the input and create the output executable, and it will execute this command for you. So this is an example of a domain-specific language. So it's a specific language just for creating workflows, for specifying inputs and outputs. And it's declarative rather than, for example, imperative. So if you're running Python, or if you're running a shell script, you're typically using an imperative type of language, which says I want to do A, B, C, D, E, and so forth. But in a declarative language, we say instead, I want to create this file, and here is how I'm going to. Here's how this file is created from some other files, and you figure out. You tell the program, you figure out how to create this file. So it's a declarative language. And from these simple rules, you can start developing really complex workflows that are nevertheless clear and reproducible. And so that's the general idea behind this whole set of the Make family of software. However, there are some problems with GNU Make in its original implementation. The biggest problem is that it's not really a full-fledged programming language. So if you have some simple inputs and outputs, it's quite good at this and quite terse. But if you want to do anything more complicated, there's just a very simple sort of programming paradigms that you can use. So there's a very simple for loop that, for example, here, and there's some way to create functions, but it's very messy because you need to call the function and then evaluate it. So this is actually a very good example of a make file. So this is actually written by Charlotte Sonneson in the Mark Robinson lab in Switzerland. But nevertheless, it becomes very clunky very quickly, and it becomes very difficult. So I wrestled with this throughout my grad school, and I decided that I'm never going back. And yeah, so Snakemake solves a lot of problems with this. So here's the same program, written in Snakemake. So you can see a few differences. First, you can see that these are essentially strings. So everything is just a Python object. So I assume that that most of you have now some basic understanding of Python, since there was a previous NGS seminar on Python. So you can see that these are simple objects. This is a list of strings. Here's another string, and here's another string which specifies the command to run. And now we can also see some additional features which make it readable. So these things in curly braces will get automatically expanded to the output and the input for this particular rule. And they're parsed with normal Python string formatting rules, if you're familiar with that. And so this is clear, so it doesn't rely on any special symbols. And but most importantly, it is all just basic Python. So everything in Snakemake is essentially just Python, and you can access variables. You can do computation in exactly the same way that you'd run a Python script, almost exactly. And similarly, Snakemake is declarative. So here we can specify a rule all. And so this is essentially saying we want to make the file hello make, and you need to figure out how to do it. So the way you make hello make is to run rule hello make, and then it'll compile it. So as I said, this is one of the really nice features of Snakemake, is that you can always just drop into Python when things get hairy. So I can run import OS, for example. In this, I'm not actually using the OS module in this particular role, but you could imagine that you could read, find paths of certain directories, or read the contents of a directory. And in fact, that's what I'm doing here. So this rule is this particular workflow now, if I can walk you through it, is essentially concatenating two files from input data and concatenating them to an output file called concatenated.txt. And you can follow along in this exercises / exercise1/snake file in the GitHub repository. So let's walk through it. So we're in a normal Python interpreter at the top here. And we can now use, we can say our input data has some pattern that looks like this. This ID is going to correspond to some wildcard called file1 and file2. And we're going to find the IDs matching this pattern. So this is just some syntactical sugar that Snakemake includes. And it's going to return these two strings, file1, file2. Next, we want to declare these two files as input to the rule concatenate, which is just going to concatenate these two files to the output. Next, we can use another special Snakemake function called expand, which essentially just creates a list of strings from a given pattern. So this pattern here is the string which has one wildcard. And this wildcard ID is going to be expanded using the IDs that are globbed using this glob wildcards function. And then we're going to just cat file1 and file2 to concatenated.txt. And additionally, there's a nice feature, which is that now there's a special variable here, called rules. And rules contains all the information that is present in this rule. So this rules.concatenate has some output, which is the string output /concatenated.txt. You can then access this programmatically in the rule all. So instead of having to type out the string twice, I can just refer to this variable and it'll automatically get the output of the string. So now I want to execute this. So I just have the example here. I'll also do some live coding shortly. But this essentially allows you to run this command. So Snakemake print shell commands is essentially telling you to print the commands to standard error. So you can see the commands that are being run. So we're now concatenating file 1 and file 2 to this output file, and then it runs it. It gives you a summary of the jobs that it's going to run. It will just run one instance of the rule concatenate to create one instance of rule all, and then it does this and it's done, and you get the concatenated output. One minor note is that to create the rule all at the end, in order to refer to this rules object, this rule must occur later in the snake file than this other rule. And so in order to specify the target of the snake file, so a snake file could have many different rules, and how do we know which one we want to create? You just specify -- all at the end of the script, preceded by these two dashes. Not only can you code in Python outside of the rules, but you can also code within the rules as well. And so previously we used a keyword called shell to run this rule, but now we're going to use a different keyword, which is called run. And this is going to allow us to drop into the Python interpreter within the rule. So what's happening essentially is it's creating a mini script that has the scope of the snake file, and this script has access to the special variables input and output. So here what I'm doing is I'm saying create the out handle for output. So input and output here are lists of strings. So here we just refer to output 0, meaning the first element in this list of strings. And then we say for file name and input, again, a list of strings. And then for each line in the file name, we're going to print that line to out handle with some white space or just an empty space at the end. And this is going to do exactly the same thing, except more verbosely than as the cat command from the previous exercise. So this you can find in exercise 2. And this is very nice if you just want to do some quick Python scripting. So for example, if you want to, you can use like the Python SAM parser to quickly parse through some files. So filtering based on some flag, for example, or something like this. Yeah, so this is quite useful again. So Python is really everywhere in Snakemake. You can use it inside the rules and outside of the rules. And as we'll see, we can also create special scripts for Python and other programming languages, but I'll get to that in a moment. So in these two examples, we have now created one file, this concatenated file, from all of our inputs data, in this case, file 1.txt and file 2.txt. But what if we want to, instead of combining them, we want to run two jobs in parallel? And this is often the case. Let's say if we have 100 samples and we want to run some RNA-seq pipeline in them, then we want to run the same set of commands to all of them in parallel. And so now what we're going to do is something slightly different, which is to create a generic rule for processing them. And then we're only going to combine them at the end in the rule all. So this step relies on something called wildcards. And here, this thing in curly braces is now a wildcard in the input and output directives. So this essentially means match. It's behaving in a similar way as glob wildcards. It's saying match any string that matches this particular expression. So, for example, now instead of concatenating these files, we're going to process file1 .text into file1.sorted.txt and file2.txt into file2.sorted.txt. And we're going to have this wildcard called ID. Importantly, the wildcards must be consistent between the input and output files, otherwise Snakemake will complain to you. And a bit of an advanced tip for those of you who might already be familiar with Snakemake, you can also use various regular expressions in the following manner to restrict the wildcards from matching certain types of things. So as you create more and more advanced snake files, sometimes the wildcards can get a bit messy, and you only want to match certain parts of the string. So, for example, let's say this ID is only numeric, then you would remove this part and just have 0 to 9 plus, and so forth. So this is useful for constraining the wildcards. And there's also a special keyword called wildcard constraints, which allows you to constrain the wildcard specifically, globally, or per rule. So now let's say we want to take this example from exercise 3 and look at the workflow so far. We can use this special command, which generates a directed acyclic graph or DAG of the workflow. And this gets passed to a special program called dot, which can then output a PDF of your workflow. And so in this case, we can see that we're running 2 separate instances of sort instead of 1 separate instance of concatenate, and that these have the IDs of file1 and file2. So so we've now explored combining files and running files in parallel using exactly the same rule for each file. But sometimes we might want to vary the execution of a particular rule based on which sample it was. So in the simplest case, for example, we might want to map one sample which contains human DNA to a human index, and another sample which contains mouse DNA to a mouse index. Or alternatively, you might want to run the same rule several times and use a different set of parameters for each run. So for example, you might want to apply some different set of thresholds, or you might want to subsample the input file using some specific set of subsamples. And so in general, we need some way to parameterize the rules based on the files. And so there are a couple of ways to do this, and I'll discuss them now. So the first way that we can parameterize the not only the rule itself, but also which files it takes as input. We can use this thing called input functions, and we can also specify parameters using the params directive. So here is a slightly more complex snake file, which is similar in structure to the others. So it is now. So we're now looking at 2 files in exercise 4 that have file1.fastq and file2.fastq. And we want to accomplish two things with these files. The first thing is that we want to map file 1 to some human index and file 2 to some mouse index. And to be clear, these aren't real indices. I've just included empty files in the repository so that we're not taking up a ton of space. And I'm also creating some dummy mapping rule. So in reality, we would use something like star. But this shows the general structure of the snake file. So that's the first goal. And the second goal is that we want to subsample both files to either 1,000 or 10,000 reads, which is specified here in the subsampling variable. So to accomplish the first goal, we're going to create a dictionary which tells us, which maps, okay, this file 1 directive to the human and file 2 to the mouse index. Then we're going to use an input function, which is here. So this index equals lambda wildcards, sample dict wildcard.id. What this is essentially doing is it's saying, I want to create an anonymous function which takes one set of wildcards, the wildcards object, as the input. And then it's going to return something. In this case, it's going to be looking up this wildcard.id and then returning index/human.index and so forth. So this is the index. This is how we're going to dynamically choose which file to use as input based on this wildcard. Right, and so this can also be a named function. So we can say like define some function which takes wildcards and return sample dict wc.id. But here we're just choosing to use a lambda function for brevity. So we don't need to name the function. We can just apply it inline. Then to accomplish goal 2, which is to subsample the reads, we can use the params directive. So params is generally useful for specifying parameters for the pipeline. And here we're simply going to take the second wildcard, which I've introduced, which is the subsample wildcard, which is going to be either 1,000 or 10,000. And it's going to just run this subsample command that this dummy mapper might have with this number. And so this gets expanded out using params.subsample. And yeah, so here, so this subsample was introduced first as a wildcard. Then we need to declare that we want to create the output files corresponding to both IDs with both subsamples. And so to do this, we again use our special expand function, which returns a list of 4 different possibilities. So file 1 and file 2 corresponding to subsamplings of 1,000 or 10,000. And another feature of this little dummy script is that we can also name files and refer to them in the shell command. So here I've named the FASTQ file as FASTQ and the index as input.index. And this gets referred to as input.fastq and input.index. And then params similarly gets this subsample variable, which can be referred to using params.subsample. And so so what actually is happening here is something called lexical scoping. So these in the domain-specific language, what we're doing is we're essentially creating objects called input, output, and params. These objects have attributes called FASTQ and index and so forth. And so what we're just doing is we're using Python string formatting to expand these objects into strings. And so there's no magic really happening. It's just Python string formatting of objects essentially. So just to clarify this a bit further, there are not only the input, output, and params, but there's also other special variables that get created. So these include wildcards, log, threads, resources, and config. So all of these special variables will get passed to the rule as we want to execute it. So here, for example, instead of using the params, I can also just access the wildcards variable directly, which is passed as one of these special objects to the shell directive. And then this object gets formatted using normal Python string formatting. So this is just a sort of explaining how this this lexical scoping is working, and it can come in handy. So now, just to show the workflow for this dummy mapper that we've created, we can see that as we said, we're now running 4 mappings in parallel. So we're subsampling 1,000 and 10,000 for each of file 1 and file 2. And it's not shown here, but the input files for file 1 and file 2 will also differ based on this index this dictionary that we've created. So going back to this dictionary, oftentimes there are many different metadata that you want to keep for different samples, and so keeping them in a Python dictionary can become cumbersome. So one way to reduce the complexity, instead of having some very complicated dictionary in your snake file, you just separate it out to a separate config file. So here there's a special keyword called config file, and here we can specify that now our information about these sample indices is going to come from this config file. And what happens in the background is that this config file, this YAML file, will get parsed to a special variable, one of these special variables again called config. And then we can access it in the same way that we would a normal Python dictionary. So sample index, and then file 1. So this is a dictionary of dictionaries. So sample index file 1 corresponds to the index that we want. So this is just one way to keep your workflows clean, basically. And we can also, in addition to using this special config file directive, we can also specify the config file on the command line, or we can even specify individual variables using this myvar equals foo, for example, syntax. So there are a lot of different ways that you can configure the pipeline dynamically. So actually, what I do in practice, oftentimes when I'm running a lot of samples in parallel, is I actually just create a separate set of TSV or CSV files for the samples, and then I parse them in using pandas. So pandas is something similar to R. It's very easy to work with data frames. In Python, it's one of the staple Pythonic biostatistic or just general statistic packages. And so just as a comment, a lot of the established Snakemake workflows, such as this RNA-seq workflow, use patterns such as this to get data frame, which you can then look up using Pandas. So really, again, we're really harnessing all the power of Python to make our workflow as tidy as possible. So I mentioned previously that it's also possible to modularize the pipeline by creating separate scripts in case, for example, a run statement gets too complicated. So you can create a set of scripts that can live in a scripts directory. And there's a nice feature when using scripts with Snakemake. And this feature is that when you're using this script directive, you get a special variable called Snakemake, which is made available to the script. So if you're running in Python, for example, you can get this Snakemake variable, and this variable contains all of the special variables that would normally be available to a shell command or a run command. So that's input, output, params, wildcards, logs, and so forth. So for example, if we're now in the script and we want to access the input, the first input file, we would do it using this syntax. And if our script is an R script, we would use, so in R, now the Snakemake is an S4 object. So we can access it using this at syntax to access individual attributes of the S4 object. And because Python uses zero-based indexing and R uses one-based indexing, then these are different numbers, but they're both referring to the first input file. So here is another example of a very simple rule, which will be running in parallel. But here, instead of using run or shell, we're going to use the script directive. So again, this will contain all the special Snakemake variables, such as input, output, and so forth. In this case, just input and output, and also wildcards, I guess. Yeah, so now let's take a look at what the script might look like. So here, we're just going to do something very simple. So this is just a script that says, I am processing file blah. And we're going to print this to the output. So we're printing to the output. And we're also going to print to standard error. So this sys.standard error. And we're going to do it for both these handles. And we're going to say, I am processing file blah. And what is file blah? File blah is corresponding to wildcards.sample. So we saw previously that we have some wildcard in the input and output. And this is the sample wildcard. And so we can access this dynamically using wildcards, snakemake.wildcards.sample. So all of these special variables are available to the script, which is quite handy. And so now I can execute this using Snakemake, and it will print also to standard error because I told it to. And it says I am processing file sample2, or I am processing file sample1. And this depends on the wildcard, which it picks up appropriately. Are there any questions? I don't know. I want to make this a bit interactive, if possible, but I'm also in the presenter mode. So let me know if you have any questions in the YouTube chat, and then maybe someone can chime in. Okay. Anyways, if you have questions, just type them, and I'll be happy to answer them. So another important feature of any workflow engine is essentially specifying the computational environment. And so this is important because for reproducible research, because if you don't have exactly the same, for example, executables or the same version of software, then it's impossible to reproduce a particular workflow. And since Johannes is also the lead developer of Bioconda, which is this easy-to-use package manager of bioinformatics software, Snakemake works exceptionally well with Conda and with Bioconda. And just as a side note, now Mamba is which can be installed using Conda actually is a new alternative that is a bit faster than Conda, so you can also use this. But Snakemake and Conda work very, very well together, and I encourage you to use this, especially for prototyping new pipelines. When you are finished with your pipeline and want to be absolutely sure that this pipeline can be reproduced down to the machine level, then you need containerization. So as you may have heard in some of the previous seminars, there are some softwares such as Singularity or Docker, which can allow the entire operating system to be packaged up into a container, and this allows for a sort of a machine level reproducibility. And Snakemake also supports using both of these environments. And actually, what I would encourage you to do in the end is actually to combine them, so you can run Conda within the context of a Singularity environment. And so this allows you both to access the ease of use of using Conda with Snakemake with the reproducibility of a container. And the way that we can do this is specified in this exercise 7, which is in the GitHub repository. And we can do this by using the special container directive. Here, we're just referring to the latest mini-conda Docker container from Continuum IMO. When you're developing an actual pipeline, you would want to parameterize this with the version, but for now, this is sufficient. And then we can also use a special conda keyword for the rule, which tells you create a conda environment with the following dependencies. So the nice thing about conda, which is why it's been so widely used, is that simple text files can be used to specify the dependencies. So this is the simplest version. So it tells you look up these two packages in the following channel on anaconda.org, I think. And you can also add version information to this YAML file, which tells you pick up exactly this version so that it's reproducible in the future. But again, for simplicity, I'm just omitting that for now. And so now we have a OS level containerization, as well as the conda level ease of use, and you can run that using Snakemake, use Singularity, use Conda. So again, using these many different options that are available on the Snakemake command line. So this particular exercise you might have trouble running if you haven't installed Singularity, but you should be able to install it using Conda as well, separately, if you would like. So you just need to make sure that the environment that you're in is able to use Singularity. And as another note, we can also create. So this is just a very simple channel, but let's say we want to recreate a very complex Conda environment with hundreds of different executables. Then we can use a special command conda env export, which will create a YAML file from a particular environment. Another way of compartmentalizing software is to use Snakemake wrappers. which also help make the rule a bit more succinct. So each wrapper contains both a conda environment, which specifies the execution environment of this executable, and it also lets you omit most of the sort of boilerplate shell command. So it allows you to just, for example, create a rule with the input and output and some parameters, and then the wrapper will take care of the execution. And there's a whole huge repository of different wrappers for commonly used bioinformatics software, which is available here. And I encourage you to also just add your own. They're very easy to write. And Johannes and the Snakemake community would be very happy to have your contribution. So for example, what this looks like in practice is we can now this is so until now these have been sort of dummy rules or something, but this is actually using SAM tools, which is specified in this SAM tools wrapper, and it's going to, in this case, just do something very simple, which is to create a BAM file from a SAM file, which we can use using the -b parameter if you have used SAM tools in the past. And so so as you know, note now, there's no run command, there's no shell command. It's all encapsulated in this wrapper. And again, be sure to use the use conda option when using the wrappers. So it'll install the this version of the SAM tools for you. So what is the wrapper actually? It's just a short Python script. So this is what it looks like in practice. So for SAM tools view, it's extremely simple. It's just running SAM tools view with a set of params from input to output, and you can only run it on one file. However, for more complex software, wrappers can become quite important. So for example, here is a wrapper for running star in the, actually this is not the wrapper itself, this is a rule using the wrapper, but you can see that there's a number of input files that are required. There's output files, and there's some set of required params, for example, the index of the star mapper and the threads. And then the wrapper takes care of the rest. So I'm not going to show the source code of this, but it's a bit more involved. And this wrapper takes care of passing the input to the output in a sort of non-messy manner.
So maybe, can you hear me now, David?
Yes,
okay,
I can.
There's a question that I was trying to, there's a question on Discord on the Snakemake channel from Mystic Mike. Can you give us a background about what you mean by running samples? Is this a pipeline for some software to use those samples? I think it was way back with the rules and samples. So if you can give your thoughts on saying
that. Thanks for drawing my attention to that. Sorry, I'm in the presenter mode, so it's a bit difficult for me to see the Discord. So can you give us, so the question was, can you give us a background about what you mean by running samples? So this is essentially we might imagine that we want to run 100 FASTQ files corresponding to 100 different RNA samples that we've collected the RNA from. So let's say we're pure bioinformaticians, and our wet lab biologists have created 100 libraries of some interesting data sets. So for example, recently, actually I've been working on SARS recently, and so we got a time course of some RNA-seq data from SARS infections of human lung epithelial cells, and I used Snakemake to parallelize running the mapping and the gene quantification and so forth on each of the samples in parallel. And so this is really what the software really excels at. Yeah, I hope that answers your question.
There's another one on YouTube from Rihanna. Is the shell script that you have set a bash script or not?
Yes, I think it actually defaults to bash. So yeah, so what happens in the background? So let's go back to right. So this shell directive, for example. So what actually is happening here is when the snake file is parsed, it creates a little dummy file, like a dummy shell script that lives in the .snakemake directory, and then this file is sort of filled in with the input and output and wildcards and so forth, and then this shell script is executed. So, and yeah, I think it's using bash by default. I could be wrong about that, but you can also specify the shell that you want to use using some of the command line options. So I think there's a special shell variable within the snake file, and there's also a shell underscore prefix variable or directive that allows you to, for example, prepend some strings to the beginning of your file. So this is one of the things that is easily configurable if you want to use your favorite shell. Okay, and yeah, you're welcome, Mike. And let me know. Yeah, sorry. Please, moderators, please butt in if there are any more questions. I would be happy to answer them. So this is all well and good. So as Mystic Mike was mentioning, this is a pipeline for running many different samples in parallel. But the question is now, how can we actually get Snakemake to run many different samples at the same time? Because you don't want to wait until, you don't want to run them serially. You don't want to run them one at a time. You want to use all the available computing resources that are available to you so that you'll get your results tomorrow instead of a week from now, for example. So there are many different ways to parallelize the workflow, and Snakemake will take care of the parallelization for you. At the simplest level, you can parallelize your workflow on your local machine. So nowadays, most computers have multiple cores, and you can use these cores to run processes in parallel. So each job will get some number of cores. By default, this is 1. But if you use this threads directive, you can specify that, for example, your star mapper might want to use 24 threads. Mystic Mike asks, I always use Python standard multiprocessing to parallelize my workflows. What are the advantages of using Snakemake? So I guess there are 2 advantages, I would say. The first advantage is just ease of use. So we can just use this threads directive to specify however, I guess it's also sort of accomplishing 2 different things. So multiprocessing, the multiprocessing module would let you use many threads for one particular script. So let's say you want to run a script, one particular script, and use many cores for that script. Then I would still use the multiprocessing module, and then I would say, okay, this script is now going to use these 8 threads or however many threads you want to give it, and then you can use this as a parameter here. So in this example, which I'm showing you here, again, it's a very simple example, but the default Unix sort takes a parallel argument, which allows you to to specify the number of threads for multiprocessing. And so this rule will now allow you to say, okay, I want to use 8 threads for this rule, and it will take advantage of this parallelization for you. And again, this threads keyword is again one of these special variables that's available to the shell or run or script command. So so it is always available to you. One side note. So again, this was specified on the command line. This tells you the total number of threads that you can use. So for example, let's say I have 3 samples that I want to sort, then I might want to use 24 cores if I have them available, using this -j, command. And if -j is less than threads, then threads will be reduced to -j. So for example, if I forget to use -j, this will still run, but only with 1 thread. So this is all well and good for your local machine, but increasingly these days, much of bioinformatics computation is done on academic compute clusters. So for example, I'm at the Max Delbrück Center in Berlin, and we have our own compute cluster called Max Cluster, and it is running a very old and not user-friendly cluster software, but nevertheless, it's using some basic. The basic idea is that it's using some cluster submission executable. Here, it's called qsub, and you can submit many jobs to this to the cluster simultaneously. And so here is an example of a command that I might use on the head node of Max Cluster to submit many jobs. So imagine that there's a Snakemake here, but these are the parameters. So here I'm saying submit with all these different parameters, and use this following cluster config file to specify the parameters for each job. So these 2 keywords are required when submitting jobs to the cluster. There are also a couple of different options for this, but I'll get into that in a moment. But what I want to make you aware of is that there's this, again, a sort of a special variable called cluster, which is expanded dynamically by Snakemake depending on the rule that is submitting this. And there's also this threads keyword, which we have specified here in the rule. So since the threads can be executed on the local machine, but some of these other parameters, such as hstack and hvmem, and so forth, and the cluster queue are specific to the cluster. So we abstract those variables out to a separate config file that we only use when submitting to the cluster. So what does this cluster config file look like? It's very simple. It's just a YAML file which contains one standard default. So this special the special keyword default means for any rule that doesn't have these variables, it will default to these. And then for specific rules that might require more memory or more heap stack or whatever, you can then request more memory for those rules in particular, and it'll inherit the other attributes from default. So, for example, here we're saying submit to the all queue, which in MDC is just the normal queue, but maybe you have a user specific or group specific queue. So and then this queue will be inherited by this particular Bismarck single end rule, as well as these modified increased memory and so forth. So yeah, so this is very clean design, in my opinion, because it allows you to keep the threads, which is general to whether you're running a job on the local machine or the cluster, as well as the cluster-specific threads, cluster-specific parameters, and you keep these separate in the separate file. And if you want to change the threads, for example, you can also add a separate threads variable here, and then here we would just, instead of saying threads, we would say cluster.threads or something like that. So yeah, everything is very modularized with Snakemake, which I appreciate. There are a couple of other options for submitting jobs to the cluster. And I'll point to two useful ones in particular. The first is this drama option, which is Drama is basically a high-level API for clusters. So many different cluster implementations implement Drama, and if your cluster does implement it, and you have the Drama bindings installed, then you can I won't get into it, but the execution is a bit more robust, and you can cancel processes by sending termination signals. Otherwise, the syntax is very similar to using the cluster directive. More recently, Johannes has, and this is actually what I would encourage you to do if you're starting from scratch, is to use this profile directive. And so Johannes has created a repository of Snakemake profiles, which contains a lot of the different job submission parameters for commonly used architectures and generalizes them. So, for example, this hvmem variable, which I spoke of previously, might be called, which is here, might be called something else on a different type of cluster, like on Slurm. So what this does is it generalizes all of these into one profile, and then you can execute it using the profile that is specific for your architecture. So nowadays, for example, Slurm is one of the most popular cluster executors, and there's a profile for that. So you can then create a more simple cluster configuration using this. And feel free to, and the nice thing again is that this is all on GitHub, and you can submit pull requests and contribute. It's a very open community. The third option, which I will not talk about in too much detail because I think most of the academic users will not be too interested in this because it costs money. But maybe the industry people are interested in, is using these different cloud platforms for running code. So now, actually, when I first started using Snakemake, none of these were available. These have only just become available in the last 1 or 2 years, I think. And so they're various different options for running on different cloud architectures. So I think the most popular architecture used in the industry is probably AWS. So this allows you to rent. So all of these actually basically allow you to rent compute nodes or compute time using some generic API. So that is specific for the type of architecture. But the nice thing is that Snakemake has options for all of these. They're a bit more difficult to get set up, so I'm not going to get into it here. But I encourage you to look at these 2 links if this is something that you're interested in. In particular, I think Google has actually given some small funding to Johannes to develop the Google Cloud interface, and so this is quite well documented. And so if you are interested in renting a large number of nodes to do your compute, I would encourage you to use the Google Life Sciences option, since it's quite well documented, and now there are these tutorials that are available here for getting set up with the Google Cloud, the Google Life Sciences Cloud. And finally, if you're more of a hacker, there's this Kubernetes execution, which is sort of the generic API for cloud, or I guess for clusters actually in general, but many of the clouds use this underlying API. So this is sort of like drama for the cloud, one might say. So yeah, so there are very, very many options for parallelizing your code. You can either do it locally or on a compute cluster or in the cloud, and all of the support is there for you. So now we've covered a lot of the different features of Snakemake. What happens when something goes wrong? And so when you're getting started with Snakemake, you might get frustrated that some code is not behaving as you expect it, or the wildcards aren't matching exactly how you'd expect it, and so you need to debug your snake file. So there are a couple of ways to do this. The first and most simple thing, which I'll point out, is that it's always useful to have a log file for your rule. So this log directive is, again, one of these special directives that gets passed to a special variable within the rule. And it just tells you, it essentially captures all standard error from the rule and passes it to this text file. Or you can also you can write to it explicitly, as I'm doing here. So that's one thing. And then the next thing that is useful is to use the Python debugger. And you can do this by default, actually, in the run directives. And you can also, there's a special debug argument that you can pass to Snakemake to allow you to run it for scripts as well. So, for example, here, what I'm doing is, so this is an incorrect rule. If you can take a moment and look at what is wrong with this rule, what's the bug? Can you find it? I'll just wait a moment before moving on. Nope, indentation is fine actually. Although, yeah, maybe. No, is there any place where indentation is wrong? I don't think so. Run is empty? No, run is not empty. Run is populated with this script. The thing that's wrong is this myVarus3. So we've forgotten that that Python is using zero-based indexing, and so we're trying to refer to the fourth element of a 3-element list. So yeah, so we when we run this, we get some. Whoops. Right, we get some error. My index error list index is out of range. But you can drop into the Python debugger when you specify this debug on the command line, and then you can go through your. You can examine, for example, this myVars element, and you can say, "Oh no, myVars3 is out of range, but myVars2 is in range," and so that's the one that is correct. So you can always use the Python debugger to examine variables within a snake file, and this is quite useful. And you can do it in the run, or also in the Python. If you're using Python script, you can do this as well. And so now we fixed it, and we're running the correct rule, and this is going to work fine. Another option, which I'm not going to show the output for, but is generally useful, is using this verbose option. And so this essentially walks through all of the underlying Python steps within the snake file. So you can often debug snake files using the verbose option as well. So several strategies to get started with debugging. Actually, so I didn't include it in the presentation, but I also wanted to mention that there's an option called debug DAG. So one thing that can sometimes be confusing is how the wildcards are interacting with each other. So for example, if you have a very generic rule, for example, like samtools view that takes some SAM file and creates a BAM file. Sometimes if you don't specify the wildcards very explicitly, it can pick up the wrong, like it can go through the workflow in the wrong way that you might not expect it. And so this debug DAG, wildcards are referring to external files, right? No, wildcards are actually referring to these strings. So yes and no. I guess I need to understand the question better. So wildcards are, so for example, these are the two wildcards. So so in this case, this general pattern of a file matches whatever the sample IDs are. In this case, I think it's file1 in and file2 in. And so these wildcards are then strings, file1 and file2, and they get passed on to the wildcards object. But the problem is sometimes this wildcard is too generic and it captures the wrong string that you might expect, and then it says there's no file there or something like this, and you need to debug this, and you can use the debug DAG function to do this. So I didn't include that in the presentation, but I think it's just worth noting generally. So those are a few strategies to get started with debugging. Next, I will talk about some nice features for creating reports or outputs from the pipeline. So now we've got a working pipeline. It's been debugged and everything, and we have a set of outputs. So for example, we have for an RNA-seq pipeline, we might have some differential expression figures, and we want to summarize these in some report that we can give to our PI, who will be then happy with us, and so forth, which is our goal in life. Don't forget. So now, recently, it's been, as of, I think, just a few months ago, actually, it's been possible to integrate Jupyter notebooks directly into the pipeline itself, which is nice because it enables a rapid and reproducible production of reports from a pipeline. And again, in a similar way that this special Snakemake variable was viewable to the scripts, the Snakemake variable is also viewable to the IPython notebook. So you can run the IPython notebook on many different files and produce a report, for example, for each sample if you want, and then aggregate them in some way. So Johannes actually made a nice demonstration of this, so I'm going to just use his demonstration. But in a nutshell, we're going to use the edit notebook option for Snakemake to create a Jupyter notebook on the fly. And you can also follow along with exercise 11 in the GitHub repository. Also, as a separate note, if you're working remotely, so often your Jupyter notebook is on a separate server, then it's useful to use port forwarding, and you can use this syntax here to specify the port that you want to forward, that you will eventually forward when using SSH port forwarding. So so this is Johannes's example of running or creating a Jupyter notebook on the fly for a particular rule. So like me, Johannes likes creating very simple examples, like rule hello with some test.txt. So now, as you can see, we are creating this notebook directive, which is another one of these special directives for the snake file. And we also need to create some environment file, which I've shown you previously. So this is just going to say we're going to execute this rule within an environment, a content environment that contains Jupyter. And we'll now run it using useconda, like I mentioned previously. Recent versions of Snakemake require you to specify the number of cores, which is a bit annoying, but I guess explicit is better than implicit. And so now we use this edit notebook parameter, and it'll open up a notebook for you automatically if you're on a local machine, or you can port forward it if you're remote. And I'll just pause it here. So here again, just like with the scripts, we have the special variable Snakemake, where we can access the output variable. In this case, that was test.txt. And we'll just use this to execute the snake file. And so we're just going to print hello world to the output file. Keep in mind that you also need to actually run this cell, which essentially extends the path to your environment. Yeah. Then you can close. Then you can run it and close it, and it will then finish the step. And now you can see. Whoops. You can see that now this test.txt has been successfully created by the IPython notebook, and we also have the IPython notebook available for us that we can generalize to other samples as well if we want to in the future. So this is the latest feature for working with IPython notebooks with Snakemake, and it's very handy. Sorry. There's also a more generic reports function, which essentially allows you to collect files that are created, such as figures, and collect them into a report, which is formatted using this RST, or restructured text. So this is just like a how do you call it? One of these mini a language similar to Markdown, except it's mostly used in the Python community, and it allows you to create captions with embedded variables and so forth. I'm not going to get into it, but you can read about it more in this report URL, and also you can look at this documentation as well. But the important thing to note is that the output files that are going to be included in the report must be embedded with this report function. So instead of output some file, you would say output report some file in braces. And so now I'm going to take a quick pause from the presentation to show you what a mature report might look like. So here is one of the more complex reports from that Johannes has created. So first of all, it gives you an overview of all the steps that have been executed. It's a bit hard to see, but there are sort of arrows. So this is the DAG that is connecting all the different steps, and you can visualize the execution of the pipeline in this way. It also gives you statistics about how long each step has taken. So here's the runtimes of each of the different steps. This can help you, debug if some particular step is taking too short or too long, then this will help you. And you can also look at the configuration file that was used to run a particular data set. So again, this is the config file. This is a more advanced config file that might be used in a real world scenario. It contains each of, so for example, here, this might be one sample, which has a tumor and normal matched DAM files. And yeah, so there are many different parameters in this configuration. And then there are different results that are specified using this report directory, this report function. And you can include SVG files or PDF files or whatever you want. And it will tell you, for example, here it looks like they're comparing different methods for estimating allele frequency. And yeah, so I encourage you to take a look at this. There's also the corresponding code, which you can find in the documentation for this if you're interested in this. But this is just to show you what this is capable of. And then next, again, we'll dive out of the presentation to take a look at one of these best practice workflows, which contains a lot of the rules that one might use when doing a normal star and a mapping followed by differential expression. This is actually something that I've extended in the past, and I quite like it. So let's take a look. So there are a couple of things here. So first of all, this is in the Snakemake workflows directory. There are also a number of other generic pipelines. So if you want to do GATK variant calling, for example, or if you want to use Callisto and Sleuth instead of Star, or if you want to do single cell RNA-seq, or single cell drop-seq, or something else, there are a number of different high-quality workflows in this directory. And again, you can always add your own. Can you please zoom in on the web page? I will try. Is that better? Good. So where am I? I'm here. So let's take a look first at the snake file, which is the master file. So we can see that we're going to use pandas. Again, this generic R in Python kind of programming module. And we're going to read in our samples into a specific data frame object, as well as our units, which are essentially more metadata about the samples. And at the top, we are using our declarative syntax to say first exactly what we want to create, and then the pipeline will figure out the rest. So here we want to create differential expression results for each contrast, which is specified in this differential expression contrasts. And we also want to create a PCA, and we also want to create a multi-QC report. And we also want to create a report at the end, which contains all the general information for this particular workflow. And then we want to run this within our Singularity environment to ensure reproducibility. And finally, we want to include a number of these rules using the include module, which allows you to modularize your code and organize it based on which functions they're doing, which again leads to a cleaner pipeline. So there are two things that we can look at here. The first thing is let's take a look at the config file and the sample and units. And so I've sort of adopted this this for all my workflows basically. But what I do is I usually have, actually I just usually have this samples.tsv, which is you can think of as a tidy data frame containing all of the metadata that you could possibly want about your sample. In this case, this just tells us where the FASTQ files are located. So they might be located outside of this GitHub repository. It also tells you the strandedness, so which type of RNA-seq library is created. But it allows you to add arbitrary columns. So I often will specify the index as well, and many other variables per sample. Then it also, you can have some specific parameters for trimming. So you need to trim the adapter. You can give this the sequence of your adapter. If it's different from the default, you can give it the path to the star index in case you're always running the same star index. And then here are the contrasts for the differential expression. So DSeq takes contrasts. So for example, controls versus infected, for example, or treated versus untreated. So you want to compare all the treated samples with all the untreated samples, or you might want to do additional contrasts, but these can be specified with this contrasts variable. Then we can take a look at samples. So again, this now contains our metadata about each sample. In this case, it's just the condition, which is treated or untreated. And then there's also the units, which contain the information about where the files are located and maybe some additional meta information about which lane it was run on and so forth. I usually combine these into one file, but here they're separate. Now we can take a look at some of the rules in depth. So for example, let's take a look at the first step, which is alignment. So you can see, first of all, this is, you can think of as a more advanced version of the lambda input function that I mentioned earlier in the presentation. So this getFASTQ function is taking a single object, the wildcards object, as its input parameter. And based on the input parameter, it is going to determine whether the input FASTQs are single end, which would then mean it's just one FASTQ file, or paired end, in which case it's two FASTQ files, as well as its gzipped and a few other things. So based on this, we can then, on this complex behavior, we can then simplify the rule and say a sample is just going to be the output of this function, which takes these two wildcards as input, sample and unit, where sample and unit are defined in the sample and unit files. And regardless of whether it's a single end or paired end, we will always create a single BAM file and a single reads per gene file if we're using star to to quantify our counts per gene or per transcript. So you can see that this is created because we're using the quantimo gene count, which creates this file. And we also give it a GTF file which contains the metadata for the RNA-seq. Furthermore, we want a lot of threads for this because star can use a lot of threads to increase speed quite dramatically. And we also specify that we want to use the star align wrapper, which is in the wrappers repository. So all of this makes this rule, which otherwise might be quite complicated, to something fairly palatable and allows you to parameterize things using the index, and it gives you a log as well. So all of this is pretty succinct for one alignment job on many files. Next, actually, there are a few other things before that. So there's QC. This here, we're using the Conda directive to specify instead of the wrapper, because there's no wrapper for this particular script. But this script is here, this GTF to bed script, which converts, of course, the GTF to a bed file. And again, we can take a look at the script, and we can see that it's using all of the Snakemake input and output files and so forth to keep it very succinct without having to add all this boilerplate. So this is just creating a DB of a particular GTF file, and then creating a bed file by hand, essentially by parsing it in some way. Yeah, and there are a couple of other functions for running. So let's take a look at, for example, deseq. So now we are in R rather than Python, because deseq is written in R. And we can say that we want to, again, we have the special Snakemake variable that's accessible to us. We can access the log file and write messages to the log file to debug if necessary. And then we can access other threads, attributes of the Snakemake variable, such as threads, to parallelize deseq if we want. And we can pass the contrasts that were passed in using this params to automatically provide the differential expression contrasts that we want. And then this is just a deseq in a nutshell, and then we will create our plot, our MA plot at the end, and it'll create this automatically. So that's just a quick nutshell of how this is working. And we can look at the some sample output, I think, of this somewhere. Where is it exactly? I think it's here. Doesn't it get complicated to debug the workflow? How does it display error messages across all supported platforms? Is the question. The answer is yes. It can be a bit complicated, to be honest. And so it's a bit of a. So I think when you're creating a new workflow, there are a few steps that are there are a few development practices that I like to take when developing it from scratch. So the first thing is I like to create it step by step. So I typically what I usually do is I create a very the structure of the workflow with just dummy information, and I first check that the structure is going to work. Then I fill in the code from the beginning until the end. So yeah, you can imagine. I say, okay, I have the general idea. I want to run star. I want to run feature counts, and then I want to run some script that's going to collect all the counts and then run differential expression on that count matrix or something like this. So that's the general structure. Then I fill in the code one by one. Yeah, and this will help in the development practice. Yeah, and then there's a special feature which actually I implemented a few years ago, which is called Until, and it basically lets you run the pipeline until a certain rule is reached. And so I find that this is very handy for doing this step-by-step coding. And so the nice thing is that you're coding and writing the pipeline at the same time. So it's sort of, so at the end of the day, you're going to have a pipeline that is doing exactly the analysis that you want. So it's sort of, I do also my exploratory coding, even using Snakemake by writing simple rules. And I'll show you, if we have time, some example of this as well. Okay, I've downloaded the report. Let's see if we can open it. Yep. So now we have some PCAs of our groups. So these are dummy files, so they're not that interesting. But we can look at an MA plot of the log fold change of one condition versus the other. We can look at some statistics of how long each rule is taken. And we can also examine the rules individually, as well as the Conda environment or their singularity environment and so forth. So, and again, this was used, this was specified, I should again mention, using this report keyword. So for example, if we go back to rules and we look at, I think, differential expression. Yeah, we can see that, for example, this PCA output is going to be encapsulated in this report function, which essentially tells you to include this in the final report. So it's very, very clear. Yeah. So that is a real-world example of a complex Snakemake workflow that is nevertheless, I think, very, very succinct and modularized. And the nice thing also about using these Snakemake workflows is that they all have automated testing built in. So they all undergo continuous integration with Travis. So you can always be sure that it's more or less running as you expect it to. Good. So we have about 20 minutes left. So first, I'll finish up by just telling you some of my favorite keywords or parameters that I am using often when I'm doing development. So for example, we can use, so let's say this is a situation that I find myself in a lot, where my PI is, or someone, maybe my boss in industry is bugging me about some output, some interesting figure that he needs like really right now because the investors wants to buy our company or something. Then you can use this prioritize target or priority keyword on a per rule basis. So you can set this to any value between 0 and 100. And so, for example, if you really want this figure right away, then it will, if you can set it to 100, and it will be the first thing that will try to be executed. Otherwise, the DAG might meander off in some other direction, more or less randomly. But here you can prioritize the execution of a certain file. So this is sort of the most useful in the real world feature. Additionally, there are some, so just like report, you can also decorate files using some functions if you want to do things automatically. So you can use the temp decorator function to tell Snakemake that you can delete this file as soon as you don't need it anymore. So just to save space. You can also use protected in case there's some output that's extremely important that you don't want to accidentally delete. It will write protected when it's created. You can use the directory keyword to create directories, so you can't do this by default. By default, Snakemake only recognizes files, just like Make. But this is useful, for example, if, for example, FastQC is creating a whole directory worth of information, then you can use the directory to just say, okay, this directory will be created as output rather than a particular file. You can download files automatically from many, many different sources using the remote keyword. So you can download stuff from S3, Google, Dropbox, and the normal HTTPS and FTP using this remote keyword. And you can read the documentation a bit more about how to use this, but it's very handy for getting large data sets. And we've already seen previously that we can use the include keyword to modularize our snake files by theme. So we can have an alignment snake file and a QC snake file and a differential expression snake file. And this will help us organize our code in a clean manner. There are also some useful command line arguments, which I'm often using. We can use force all to just rerun everything from scratch, which happens if you've made a mistake. You can use keep going on the cluster to prevent your workflow from stopping. If a step fails, then all of the other independent steps will continue, even if that one step has failed. The default is to stop. I've already mentioned these features which I've implemented, which are until and omit from, which says basically run the pipeline until you reach a certain rule. Delete all output is the equivalent of make clean. If you've used make, it's essentially saying just delete everything that you've created and start from scratch. You can use the built-in Python or Snakemake linter to make your code pretty. And we previously created a DAG based on each individual file, but you can also create a more general rule graph using this parameter, and this will give you a more broad overview of the workflow. And finally, you can use the archive command when you have finished an analysis and want to package it up for example, for a publication, or if you want to give it to a collaborator, you can just package everything up automatically, including the Conda environment, and it will be a self-contained, reproducible pipeline. And there are something like 80 more different options for running Snakemake, so it's really a very, very feature-rich executor by now. Some other useful features that I'm not going to get into are between workflow caching. So if you have two different versions of a run that use similar intermediate files or the same intermediate files, you can cache them between workflows. You can have so-called provenance between the workflows. You can track the different parameters and code changes that you run each time, and this gets stored as special diffs in the .snakemake directory. You can pipe stuff between jobs to make it sort of more like a Unix pipeline. You can do something conditionally if the pipeline is successful or fails. You can run some custom code. You can create shadow directories in case each instance of a rule is accessing some common resource in the main directory. You can sort of create individual directories for each rule rather than running them all in the same directory. And there are many cluster submission features which are useful, since a lot of academic bioinformaticians are using clusters. So file latency, you can create dependencies on the rules. You can submit them, all the jobs in bulk simultaneously, and so forth. So there are many features, and I encourage you to explore them all. So finally, here is a set of resources which might be useful. If you have any questions, the about a particular issue that you might run into, Stack Overflow is now the official Snakemake question and answer forum. So everyone should submit questions here. Here are the wrappers. The GitHub code itself lives here. There are some nice tutorials on using Google Cloud with Snakemake, the profiles, and finally Bioconda, which Johannes and many other people in the community are heavily involved in. So we have 10 minutes, I think. Are there any questions from listeners? If not, I can also give you another real-world example, this time using a real snake file that is associated with the publication, which I think is quite nice. But I'll wait for one moment for additional questions. Maybe there's something on YouTube. Actually, I haven't been checking YouTube, so I'll do that. One moment. Okay, I actually can't see YouTube. Yeah, please speak up if there are any questions. Otherwise, I'll continue with this one more final real-world example, which I quite like. So, Hyeshik Chang is the author of this particular pipeline, and Hyeshik is a former graduate student of Nari Kim in Korea, and he's now an assistant professor at Seoul National University. And he was also one of the early adopters of Snakemake. So, he's also, we've interacted a little bit in the past, and he's also now doing similar research to us. So it's nice to follow his work. And since he's also very passionate about Snakemake and reproducibility, he has put all of his code for this recent cell paper of his on the SARS-CoV-2 transcriptome available. And so all of this is available on GitHub, and you can take a look at it as you like. Here's the URL. And essentially what this paper is doing is it is using several next generation sequencing techniques, including nanopore and also this DNA nanoball sequencing, which is essentially like a very high resolution RNA sequencing, to look at the transcriptome of SARS-CoV-2 in extremely high resolution. And so they found some interesting things by using direct RNA sequencing. They looked at the poly-A tail lengths and looked at RNA modifications. And they also found discontinuous transcription events in the transcriptome. So so the nice thing here is that all of the code for running, for example, nanopore, is present within this snake file. So what they did, in a nutshell, was to run direct RNA sequencing on an RNA sample isolated from Vero cells infected with SARS-CoV-2. And then they collect the data and base call it, and then map it to either the viral genome or the monkey genome. So Vero cells are from monkeys. And they can see some interesting things, which I'll get to in a moment. But you can see that he's creating many different files based on the viral samples or on the alignment type. And so he can specify all of this in the rule all. Here are some additional features. So in order to base call nanopore data, you need to use something called Guppy. And this is a bit tricky because you need to use a GPU. So actually, it looks like he's not using any of the GPU features. So there's some, so for example, with Google Cloud, they have a GPU resource engine, but he is not using this. Then he's going to merge all the sequences and map them to a genome. So here we're using the same genomic index, which is using Minimap. He's now using the wildcard constraints that I mentioned previously to constrain the wildcards for this rule. And another thing that you can see is that he's using some constant variables embedded within the shell scripts, like junctions. So junctions is one of these constants that's defined at the top. So here they have a bed file that contains all of the known. You need to know a bit about SARS biology to understand this, but basically SARS has some set of known splicing-like events that occur, and the known ones are contained in this bed file. So it's a very complicated snake file that is nevertheless quite clean and uses a lot of the different features that make Snakemake so nice. So that's the execution. Actually, Hiashik hasn't started using the IPython edit notebook feature yet. But what's nice is that he collects all of the figures or all the data for creating these figures in these IPython notebooks alongside the data. And I think in some other ones you can actually see some of the figures. Yeah. So here, for example, he's quantifying these so-called junctions, which are the known splice sites, which correspond to known subgenomic RNAs of the virus. And if you're interested in this, I also encourage you to read the paper. It's now published in Cell. It's called The Architecture of the SARS-CoV-2 Transcriptome. But you can really spend a lot of time going through the code that he's so kindly made available to everyone. So that I think is really reproducible research at its best, when there's a well-documented or reasonably well-documented set of code which has everything for executing the pipeline and the creating the figures alongside it. Okay, so with that, I would just like to thank many people. First of all, many thanks to Johannes Köster, who has been, over the years, not only a wonderful person to work on software with, but also a good friend, as well as the rest of the Snakemake team and the Bioconda team for creating and continuing to actively develop this really nice software. I'd also like to thank my lab, the Rajewsky lab at the Max Delbrück Center in Berlin, many of whom are also very avid Snakemake users. And lastly, I'd like to thank Kasia in particular for inviting me to give this seminar today, and to NGSchool, and to all of you for your attention. So, thank you very much.
Thank you very much, David, for today's seminar, and also for answering all of our questions. As someone who's never used the platform, this has been very informative for me, especially the real-life examples. And I see that everyone who joined us also feels the same. So thank you very much.
Okay, it's nice to hear that. Thank you very much.
All right, so this ends our seminar today. Thank you everyone for joining us. But before everyone leaves, we have some announcements. So first of all, we have extended the abstract deadline for our NGS symposium to the 27th of August, so more time for you to submit. Second, we have another seminar next week. It's about isoform detection from long-read RNA sequencing. It's a very interesting topic, so please join us again. And third, last but not least, please remember to give us feedback about the seminar on Discord. So, okay, thank you again, everyone, for joining, and see you all next week.